the time to that they were awarded. So the next speaker is Wenlei Jiang. She is Deputy Director from CBER, well, the Office of Generic Drugs, CDER. And that means she comes from the US as well. Regulatory matters, US view.
Thank you, Dan, for the introduction. Good afternoon, everyone. My name is Wenlei Jiang. I'm from Office of Research and Standards, Office of Generic Drugs, CDER, FDA. It is my great pleasure to be here today and present Assessing Equivalence of Generic Nanomaterials, FDA Perspectives. So the idea of nanomaterials was first proposed by Nobel Prize winner physicist Richard Feynman in 1959. In the past 50 years or so, and many nanomaterials have emerged, including nanotubes, polymeric micelles, nanocrystals, liposomes, and dendrimers. These nanomaterials were designed to achieve targeted drug delivery and improved drug bioavailability. And in 2014, FDA finalized its guidance considering whether an FDA-regulated product involves the application of nanotechnology. In this guidance, it was highlighted that the material need to be engineered to have approximately 1 to 100 nanometer size or up to 1 micrometer if the product exhibits unique property attributable to its dimensions. So FDA agrees that the application of nanotechnology may result in product attributes that differ from those of conventionally manufactured products, and therefore may merit further examination. However, FDA does not categorically judge all products containing nanomaterials or otherwise involving application of nanotechnology as intrinsically benign or harmful. Nanodrug product development has been very active. There is a strong product pipeline. Based on a 2012 FDA database search, there are 128 IND applications containing nanomaterials and 30 NDA, AND applications containing nanomaterials. So over 50 NDA, ANDAs containing nanomaterials were approved, and the generic approvals account for about 10%. So for oral products containing nanomaterials, FDA thinks that the generic applicants can opt not to use nanotechnologies, but other approaches such as solid dispersion or surfactants to improve bioavailability. However, for parenteral or topical products containing nanomaterials, FDA recommends that both test and reference products have equivalent particle size distribution, unless there is scientific evidence that the size is not critical to safety or efficacy. As you may know, over 80% of prescriptions in the US are filled with generic drugs, and generic drugs has saved over $1 trillion in the past decade. And generic drugs are approved based on meeting pharmaceutical equivalence and bioequivalence standards, and the approved generics are considered therapeutically equivalent and interchangeable with the reference drug. For most of the nanodrug products that are for parenteral use, there have been some unique challenges in demonstrating therapeutic equivalence of parenteral nanodrug products. Some parenteral nanodrug products have high-order supramolecular structure. For example, in the case of Doxil, it has the lipid bilayer structure and PEG coating. So a formulation with qualitative and quantitative sameness cannot ensure the higher order lipid bilayer structure sameness or PEG coating sameness. Also, some of the parental nanodrug products are designed for the target delivery. In this case, the conventional plasma PK may not fully reflect the rate and extent of drug available at the target site. And also, total drug alone, as Scott mentioned earlier, may not be sufficient to demonstrate bioequivalence. So these unique challenges have hindered the development of generic nanodrug products. And given the diversity and complexity of nanomaterials, FDA has taken a case-by-case approach to develop product-specific guidance for each nanodrug product. Here is a paradigm. First, we require ANDA formulation to be qualitative and quantitative the same as a reference drug. Next, we try to understand the mechanism of action and ask the question, what's the targeted product profile? What's the key physical chemical properties which may impact the particle uptake, distribution, and release in vivo? Based on this analysis, we recommend a comprehensive physical chemical characterization and ensure physical chemical equivalence of some key quality attributes, including particle size, distribution, surface properties, and others. And furthermore, for parental nanodrug products which can dissociate slowly in vivo, we recommend to measure both the unencapsulated and the nanoparticle bound drug to ensure equivalent systemic exposure. However, for some nanoparticles which will dissociate completely and immediately after administration, the formulation Q1 and Q2 sameness, as well as physical chemical equivalence, may be sufficient to demonstrate product equivalence. It has been said that the lack of clear regulation guidance remains to be the key bottleneck limiting generic nanodrug development. Here, I have to say, in the past several years, FDA has made significant strides in this area, providing product-specific equivalence guidance. Here is a list of nanodrug products which FDA has published its guidance, including liposomes, iron complex drug products, and paclitaxel albumin bound particles, as well as several products, generic nanodrug products approved in 2011 and 2013. Here, I would like to highlight draft guidance on doxorubicin hydrochloride. This is the first product-specific nanomaterial guidance we published. In this guidance, it lists the details of some of the physical chemical characterization tests, as well as the rationale behind. So this guidance provides clear pathway for generic doxorubicin liposome development, which finally leads to the approval of this product and alleviated doxil shortage crisis. As many of you here know, EMA does have a different opinion about the requirements for generic liposome and iron product approval. EMA emphasized the importance of non-clinical animal studies to investigate product biodistribution. However, EMA does admit that there is insufficient regulatory experiences with non-clinical biodistribution studies to recommend any specific decision criteria for the evaluation of the comparability of the tissue distribution. So realizing these controversies and the challenges in generic nanodrug development, FDA has funded a number of research studies related to nanomaterials, including studies on physical chemical characterization, non-clinical studies to evaluate the difference between FDA and EMA approach. The ultimate goal of these research studies are to support guidance development for nanomaterials and also ensure the safety and efficacy of approved generic nanodrug products. So these research studies involves FDA internal office collaboration as well as external collaboration among academia, pharma, and FDA. Next, I would like to highlight a couple of these research studies. One challenge in developing nanodrug product is to identify critical physical chemical properties which may impact the in vivo performance, and also establish the analytical technology to sufficiently characterize these properties. So we have funded studies to explore the capability of different analytical technology, including small angle X-ray scattering analysis, cryo-TEM, and others, to characterize liposome formulations that span a range of formulation composition, manufacturing process, source of materials, and particle sizes. Of course, the Doxil and its generic counterpart were included in these characterizations. So data available so far demonstrated that both brand and generic doxorubicin hydrochloride liposome injection have similar physical chemical characteristics. And also, these analytical techniques are able to distinguish them from other liposome formulations prepared in the lab. Some remaining questions are when to use and what are the quantitative criteria to compare these physical-chemical parameters. Some recent efforts in the in vitro dissolution method is to developing single vial-based in vitro method as well as USP apparatus 4 flow-through assay to analyze drug released from ambisome and doxorubicin hydrochloride liposome injection. And these newly developed methods are able to distinguish dissolution profile from formulations with similar composition but slightly different manufacturing difference. And PB pharmacokinetic modeling simulation recently had received considerable attention. Here, physiological-based absorption modeling have been explored to develop quantitative correlation between physical-chemical properties and in vivo performance. Also, PK modeling and simulation has been used to determine which drug moiety is most sensitive to detect nanomaterial formulation difference, and also predict in vivo non-transferring bound iron based on the in vitro labile iron determination. Another area we have focused on is to conduct post-marketing surveillance evaluation of approved generic nanodrug products. Patients and physicians were surveyed about their opinions on the approval process of generic nanodrug products. And also, a well-controlled prospective PK study to evaluate iron species in healthy subjects treated with generic and reference sodium ferric gluconate is ongoing. In summary, FDA recognizes the complexity of generic nanomaterials and recommends a case-by-case product-specific approach for nanomaterial evaluation. FDA proactively develops product-specific equivalence guidance to aid U.S. generic nanodrug development and approval. We are funding regulatory research in generic nanodrugs. These research studies will enhance regulatory understanding, support guidance development, ensure post-market drug safety and efficacy, as well as promote international regulation harmonization. So I hope with these efforts, we can improve patients' access to nanodrug products at an affordable cost and also build patient confidence on this advanced drug delivery system. Yeah, thank you for your attention. Yeah, I also want to thank my colleague in Office of Research Standards and external collaborators for their contribution to this project. Yeah, thank you.
Thanks. Questions for clarification? Only one. I allow only one. Because we have ample time later on to discuss. Yes, please.
There's a lot of work there about measuring the size, but I didn't see anything about measuring the number of particles, and it seems to me if you're going to characterize the formulation, you would need to know the number of particles as well as the size. Otherwise, how do you know how much drug you've got?
Yeah, actually, I didn't include the details here. We do have multiple particle size analysis methods to characterize these products, and these are done by third-party independent.
It's particle number, not particle size. Particle number is different than size.
The answer is in this book.
Okay. Did you write it? Is it your book?
And Scott also knows quite a bit.
Excellent.
About that.
Yeah, I think we know the QNano analytical tools.
There's a lot of material in there. No, I'm serious. There's a long chapter. I think the longest chapter is on particle size. Thanks, Malin, again.